Maayong buntag sa inyo ha, mga beloved students of Cebu Normal University, and I hope that you are doing well. I hope that you are doing fine. I hope that you are happy. I hope that you are in love. Kana siya. So, mao na nga, I will be happy if you are also happy. Na, Gemma, Char. But anyway, welcome to our another discussion sa to ang subject ng biotechnology, and I will be your instructor for this course. And sa karon, this will be our third nga topic for this semester. Okay, so we will now go sa topic number 3, which is recombinant DNA technology. Okay, so let me just share. Ayan. Okay, so I hope nakita na sa inyong screen. So, we now discuss this topic. So sa itong topic, we will be having three subtopics. We have the restriction enzymes, we have cloning, and the DNA library. Okay, so to recall, when you talk about biotechnology, that is to use technology in enhancing kaning gitawag nitong mga biological nga mga entities. Okay, so i-enhance nimo ang usa ka organism or i-enhance nimo ang product sa usa ka organism. So that's actually relatively biotechnology. And just to help you recall, remember that biotechnology is already practiced in the ancient times, only that it becomes more advanced sa ato ang karon. Okay, so when you talk about recombinant DNA technology, this is a specialized form of biotechnology. So by definition, recombinant DNA technology is a technique where whole DNA, take note ha, whole DNA and or just segments of this DNA, DNA segments of interest from different species are being combined. Okay, so meaning to say, you can manipulate the whole genome of an organism or just a genome of interest. Pwede nimo nga gamay lang ang imong enhance, or you can get some of these segments and then try to put them as one or assemble them as one. That is actually a recombinant DNA technology. So when you say recombinant, is it to recombine, okay, whole genomes or segments of these genomes, okay, from different species combined. So when you talk about recombinant DNA technology, this is the technique. This technique is also known as genetic engineering. So do not be confused by this one. So you can be an engineer, genetic engineer, and that's actually a cool job. Okay, so kana siya. So I think you are familiar with these movies. Say for example, the Jurassic World, kani siya Rampage. This is actually an interesting film by Dwayne Johnson. Then you have Spider-Man and you have Avatar. Okay, so all of these movies, of course, we know that they are fictional. Again, fictional siya ha, kay basin na pay mga toddlers nga mga minds nato din take note that we must be able now to distinguish which one is accurate in reality, okay, what's real and which are fictional. So these are actually fictional, okay, pero naa sila gi-base sa ilahang movie is nag-base sila sa what is happening in the sciences, only that ilahang gi-bloat, okay, artistic nga gi-bloat nila artistically aron maka-interest sa mga tao. Kita pag ganahan pud ta magtan-og mga sci-fi, science fiction nga movies, and I love watching science fiction movies. Okay, so din kani siya Jurassic World, mga dinosaurs, this one, creatures, animals become bigger, Spider-Man being beaten by mutant nga kaning spider, and of course Avatar being operated by DNA. They are actually products of genetic engineering. Okay, so when you say rDNA, sa recombinant DNA technology, rDNA stands for recombinant DNA after the successful attachment of DNA segments to another DNA. So, if makakita gani mo og naay kaning gitawag og small letter R and then DNA, meaning to say it has been genetically manipulated. Okay, genetically manipulated. And when you talk about chimeras, they are the organisms that carries rDNA or recombinant DNA. Okay, so kana siya. So at least we have now some new words nga ato na napulot karon. So dako ang kay nino ato lang ning paggamyan. kana siya. I think makita ra gyapon sa inyong screen. Okay, kana siya. So when you talk about recombinant DNA technology, genes for a particular trait are deliberately identified. When you say deliberately, meaning to say there is this intention of cutting segments of DNA and then i-cut siya and i-combine it with other desired genes. So, kana siya, copy paste or cut and paste, not copy ha, but cut and paste, di ba? So panalitan sa inyong homework na kay isulat, di ba, mangita kag source or mangita kag quotation gikan sa kanang website, okay, i-copy nimo, copy lang nimo, pwede di naman nimo cut, pero i-copy nimo and then i-paste nimo, i-integrate nimo sa imong trabaho. That's actually what is being done when you do genetic engineering, okay. Pero sa genetic engineering is i-cut yun nimo ang segment sa usa ka DNA and try to combine it with another genome or another DNA that will become the rRDNA. So say for example here, gene of interest being cut from a one source or source A, and then the vector here, which is now the source B, combine them together, you now have recombinant DNA technology. Okay, so that is deliberate, meaning to say scientists or genetic engineers have already identified what type of product nga kaning gene ang ihang ma-produce, di ba? Recall sa katong gitawag nato nga central dogma, di ba? So na transcription, translation, di ba? Katong replication, transcription, and translation. So, ang katong mga scientists, they already have an idea kung unsa ang protein nga gina-produce aning gene of interest. And then, gi-insert nila sa usa ka vector and then gi-expect nila that this organism carrying the chimera that carries this rDNA will now produce the genes or the product of the genes coming from a foreign nga source of DNA segment. Okay, so when you talk about um, recombinant DNA technology or kaning rDNA, okay, there are three methods that can be done. First is the transformation, second is the fudge introduction, and the third is non-bacterial transformation. So, just to give you a glimpse, when you say fudge, this actually pertains to viruses that infects bacteria. Okay, mo na siyang mga fudges. Bacteriophage, mo na siya. Bacteriophage, and in short, that's fudges. But anyway, we are going to discuss this one. Okay, so let's go to the first process. That's the transformation. So when you say transformation, this is a process where an organism acquires exogenous DNA. Okay, so what is this exogenous DNA? It is defined as the DNA outside the genome of concerned subject. So say for example, human DNA is inserted into bacterial DNA. Okay, the former is the exogenous DNA. So meaning to say, kung kinsa ang source sa katong gene of interest nga gi-insert siya, mautoy gitawag natong exogenous DNA. So say for example, balik ta ani kini siya. So this is now the exogenous DNA. Okay? Kay coming man siya from foreign, and then ang vector, say for example, this vector comes from the a bacterium, and then ang interest o kaning gene of interest coming from humans gi-insert din sa vector nga gi-own sa usa ka bacterium. Okay, and then kini na yun ang bacterium, kini siya, this will now be the exogenous DNA. Okay, so I hope that's clear. Okay, so the former is the exogenous DNA, so that's the human. Okay, so when you say transformation, it's here. Okay, so take a look at this one, gene to be transferred. Okay, so plasmid, when you say plasmid, now take note ha, that in many nga mga biological application na ano sila mag-work sa kaning bacterial plasmids. Okay, when you say plasmid, yeah, murag na kani. So this is a bacterium. Okay, mao ni gyud ang main nga DNA, circular DNA, the chromosome, because in bacterium there's only one circular DNA. Mo na sa mga bacteria ha, one circular DNA. Unlike sa humans, we have 46 chromosomes. Di ba? 23 pairs. Sila ha, isa lang yun ka chromosome and then circular pag yun. Sa humans are linear man. Now, aside from its circular DNA sa ihang chromosome, napa na siya gamay nga circular nga DNA that is known as plasmids. And majority of these biotechnological works are actually they are actually manipulating the plasmids, bacterial plasmid. So say for example, sa kining plasmid, take a look at this one. Lain na siyang color, lain na po siyang color na kay purple, na kay red, na kay green. Okay, so meaning to say, this is now an rDNA. Okay, kay recombinant na siya nga plasmid. So gi-insert. Okay, and by the way ha, not all bacteria carries plasmids. Okay, not all bacteria carries plasmid. So say for example, kaning gitawag na tong Agrobacterium tumefaciens. This is actually one of the model organisms, if I'm not mistaken, model organisms yeah. So Agrobacterium tumefaciens. So gi-insert ang kaning rDNA, okay? And then dire, okay, ang plant cells colonies gi-integrate siya dito, transform bacteria, introduce plasmids into plant cells. So ang bacteria iyahan ng gi gi-introduce dito sa plant cells ang kaning gitawag natong recombinant plasmid. Okay? So meaning to say, this recombinant plasmid will now be integrated into the plants. Okay? So once nga gi-integrate sa plants, dinha sulod sa cells, ang kanina dayon inside plant cell, agrobacterium inserts part of its DNA into the host cell chromosome and complete plant generated from transformed cell. So dinha na dayon motubo ang whatever crops nga inyong gi-desire nga patuboon, carrying the different genes of different organisms nga mo contribute sa growth, which is now becoming better nga crops. Okay, siguro say for example kining red is for pest resistant ni siya. Okay, ang green kini siya is drought resistant. Ang third dinhi is mas nindot ang iyang pagbunga. Okay, so gi-insert siya dinhi sa Agrobacterium tumefaciens, gi-incorporate sa Agrobacterium tumefaciens sulod sa plant cells ang kining plasmid, hangtod nga pagtubo niya, it is expected nga drought resistant, um, what else, more productive, and kaning pest resistant ang isa ka plant. Okay, so hinana siya pag-transform. So gi-transform nimo ang usa ka organism carrying now the recombinant nga DNA. Kana kung desired na nimo. Pero kung mapataka lang pod, dili mo na alkansi ka sa oras, di ba? Alkansi ka sa resources. That's why when you talk about genetic engineering, it should be deliberate. Meaning to say, kabalo ka, you have already identified what are the genes that you have to incorporate from one organism to another organism. Okay, so here, this is also another form of transformation. Okay, so here na kay DNA sa humans, for example, sa humans na kay DNA, gikuha nimo ang DNA, gi-identify nimo kung unsa na siya nga gene, okay? Gi-cut nimo, di ba? Kay possible sa katong mga restriction enzymes, especially sa CRISPR, di ba? Restriction enzymes gi-cut na nimo, gi-integrate nimo sa plasmid, okay? Ang dinhi pod sa bacterium, gikuhaan siya og segment. Okay? Kuan siya segment, gisumpay ang duha, and you have now here a recombinant nga gene. So say for example, kini sa medical biotechnology, what if this gene is producing the kaning gitawag nato og insulin, di ba? Si insulin, kani nga gene, di ba? Insulin is a protein, it's a hormone. Okay? Yeah, it's a hormone, and then it's a protein, and proteins are actually products of translation. So it still goes back to the DNA, 'di ba? So kani siya, insulin siya, gi-insert siya sa bacterium, and then ang bacterium now carrying this recombinant gene, carrying now the human gene for insulin, nagmultiply, 'di ba? Binary fission. Nagmultiply hangtod nga this bacterium will now produce human insulin. Bacteria sila ha, kani siya is bacterium sila, but they are now producing human insulin. Okay? So, meaning to say, aside from that, there are potentially other nga mga application for recombinant DNA technology. So, this is not the second, which is now the fudge introduction. So, when you say fudge introduction, this is also known as transfection, where the process is similar to transformation, only that fudge is used as vector instead of bacteria. Okay? So, mao na siya. So, na-discuss naman nato a while ago ang katong bacteria as vector. This one, similar ang ihang process pero only using this time viruses. Mao na nga, moingon ta nga viruses maka-cause yes, it can cause infection, but but we can also make friends with them and try to manipulate them. Okay, so this is now an introduction sa or the illustration of transfection. So you have here, okay, the nucleic acid sequence. This is now some of the bacterial components. Now, because take note ha, that since bacterial viral components, since viruses are much simpler compared to bacteria, there are instances that ang mga scientists can actually synthesize, quote unquote, synthesize viruses. Kay simple ram gyud kaayo if you I don't know if you have niagi na mo kuan ka ng microbiology, so comparison wise, structure wise, simple ra gyud kayo ang kaning gitawag natong viral structure. So integrate siya, gihimo siyang fudge. Okay? And then the kuan man gud kani when you talk about fudge kato kung gi-mention ganiha, fudges or bacteriophage, they infect bacteria. Okay? Bacteriophages, they are viruses that infect bacteria. So kini siya ang viruses or kana siya instead of bacteria o kani siya maoy naghatod dito ang DNA na dayon coming from kaning nucleic acid. So say for example, gikan ka sa katong nag-CRISPR ka or katong nag-restriction enzyme ka, ikat nimo, gipasulod nimo sa gipasulod nimo sa cell which is now highly engineered to instead of bacteria alone, dito na pud ka sa kaning gitawag ng eukaryotic cell. So, gi-release niya and then ang DNA can now integrate or can now manipulate the nucleus ang katong inserted DNA dinhi and then na mo undergo siya sa protein synthesis, di ba? Sa katong central dogma. So, meaning to say, you can actually deliver this nucleic acid sequence into a cell. Okay? Specific nucleic acid sequence into specific cell. And that is actually a recombinant DNA. Okay? So, kana siya, where in fact, kana siya, ato lang ni insert, some drugs are actually using this principle. Some drugs, ha, so instead of nucleic acid, ang ilang ibutang din ha, they are actually inserting certain drugs and then mo target sila og specific nga cell for that drug and then mao to nga i-utilize day na sa imong cell ang kanang drug. So that's actually drug designs, di ba? One of principles of the drug designs. Okay, so kana siya. So this is also another kuan sa kaning gitawag natog fudge. Okay, so fusogenic lysosomes. Okay? So gi-insert nimo din ha because hinani man gud ni ang principle sa viral nga infection, di ba? So mo-fuse ang iyahang kaning liposomes dinhi sa cell, di ba? Katong sa plasma membrane, okay, or the cell membrane. So mo-fuse na siya, okay? And then i-deliver niya ang specific nga mga molecules sa specific nga cell. So say for example, katong i-mention ganiya, say for example, pwede na siya nga nucleic acid or pwede po siya nga drug, ipasulod na niya, genetically engineer this one, and then fuse siya, delivering the products inside the cell. Okay. So the third here is na kay gitawag nga non-bacterial transformation. So similar to bacterial transformation, only that hosts are non-bacterial. Okay, ang host na ang non-bacterial. So DNA is directly injected into the cell. So say for example, micro-injection. So ganiha, we are using viruses, we are using bacteria to transform ang kaning gitawag nato nga specific nga organism of study. But this time, ang kini siyang non-bacterial transformation, DNA is directly injected into the cell or kanang gitawag tog micro-injection. So say for example, I don't know if you can clearly see this one. So kini siya gikuhaan og nucleus, okay, gikuhaan og nucleus and then giilisan siya og nucleus through micro-injection. Okay? Through micro-injection. And later on sa tuang siguro sa tuang medical biotechnology, we are going to see what are some of these applications sa micro-injection. Okay. So kini siya. Ay wala pa man dito magkuan na. Okay, but anyway, sa restriction enzymes, dili ta sa restriction enzymes. So inside restriction enzymes, katong restriction enzymes, they are proteins that cleave specific regions of DNA. So how come kato atong gi-mention kaniha nga how come nga nakat nato ang specific segment sa DNA? That is made possible through these restriction enzymes. So they are proteins that cleave, mean to say makakat siya specific regions of DNA. So take note that restriction enzymes are sequence specific. They produce fragments of DNA, same as restriction enzyme can cleave fragments of various, same restriction enzyme can cleave fragments of various sizes, and they are produced by bacteria. Okay, ang mga restriction enzymes. And later on, I will be discussing to you what are these restriction enzymes. But for now, there are four types: type 1, type 2, type 3, type 4, and I don't have to deal with these different four types. Now, ang types are actually based on structure, specificity, cleavage sites, and cofactors. Mao na ang pag-determine which type of kini gitawag natong restriction enzyme ang kini siyang gigamit nimo for biotechnological purposes or genetic engineering purposes. So take note that different DNA samples cut by the same restriction enzyme can be joined. So say for example, if ang usa ka naa kay duha ka DNA samples, okay, na kay duha ka DNA samples coming from different organism, the same restriction enzyme, say for example, this is A, okay, imuhang gigamitan og restriction enzyme. This is organism B or DNA B, gigamitan nimo the same nga restriction enzyme. So that restriction enzyme can cut specific fragments here, and that the same enzyme can cut different fragments here. Now take note nga this restriction enzyme, once nga mo cut siya dinhi sa kaning duha, naay uban nga makat nga mugbo, naapoy uban nga makat nga tag-as. Okay? Kana siya. So later on ato nang tan-awon how is it possible nga yun ana siya. So when you say restriction enzymes, they are the molecular scissors. Kana siya. Mura siya og kanang gunting, molecular scissors. Di ba? Kung say for example, mag-cut ka og ribbon, okay? Unsa ka taas, asa dapita, okay? You can actually choose which side. Okay? The same is true with kanang gitawag natong restriction enzymes. Pwede makapili ang mga scientists, ang mga genetic engineers unsa ka taas or unsa asang dapita ang pwede putlon sa usa ka sa usa ka genome. Okay? And at the same time, ang kining gitawag natong restriction enzymes reads palindromic sequences. When you say palindrome, mirror image siya. So, kung unsay imuhang makita sa left, mao na imuhang makita sa right. So, say for example here, kini siya nga sequence. This is now palindromic. Okay? Palindromic ni siya. So, say for example, this one and that one. Right? Okay. So, that's palindromic. So, kini po dinhi. Okay? So, kung unsay imong makita dinhi, similar po imong makita diha. Okay? So, that is actually palindromic. And I hope you have that one. You get that one. So, say for example, another one here, na kay two types of restriction enzymes. Okay? So, say for example, you have DNA template here. Na kay ang restriction enzyme, pwede siyang exonuclease. Ang another one is pwede siyang endonuclease. So, restriction enzymes that are classified as exonucleases, they excises or they cleave fragments at the terminal ends dinhi sa mga tumoy. Okay? Whereas ang endonucleases cleaves segments between terminal ends. So, naa siya definitely naa siya sa within terminal ends. So, in between terminal ends. So, that's some of the another classification of restriction enzymes. Okay. So, as I have mentioned a while ago, restriction enzymes are actually scissors. Okay? And, di ba, kato akong gi-mention ganiha, nga recombinant DNA technology is a deliberate nga action. Meaning to say, kay balo ang usa ka genetic engineer where to cut. Okay? So, mo na siya. So, say for example here, gi-cut na niya dinhi. Okay? Mao po di ang target site is gi-cut po na din ha. Now, naa kay gitawag nga sticky end. So, when you say sticky end, say for example, this is kani siya. Kini nga side here, this is organism A. Kini is organism B. Okay? Since they are using the same nga kaning gitawag nato nga restriction enzymes, okay? Nikat sila og portions nga nakaproduce og sticky end. When you say sticky end, these are the fragments that can attach, okay? Can be attached even though that they come from different sources. So once nga ma-attach na siya, kana siya, then you now have a recombinant DNA or rDNA. Okay, so gani naghisgot tag sticky end in he. Okay, so giunsa man na pag-form ang sticky end. So it's here. Okay, because when you say restriction enzyme, okay, when you say restriction enzyme, by the way, most of these restriction enzymes are endonucleases. Okay, most of these one are endonucleases. So ganihan naghisgot tag restriction enzymes. Now there are two results sa kaning restriction enzymes. It could have sticky ends or it could have blunt ends. Okay? When you say sticky ends, 'di ba? Kato akong gi-mention sa inyo ha is palindromic man, 'di ba? So kini siya is palindromic, okay? Kana siya. And then ang recognition site din ha is ni cut siya din he. Okay? Pag-cut ana niya, na kay fragments nga kini siya is makita nimo dinhi wa siya partner, ang iyang partner gibulag sila dinhi. Okay, mao ni siya kaning kini nga part nga walay contemporary strand niya, wala siya kapares sa base pair. Okay, mao ni ang gitawag natong sticky end. On the other hand, ang kaning gitawag natong blunt end, take a look at this one, it cuts perfectly sa duha ka kani into two different sides. Meaning to say, all base pairs, quote unquote, all base pairs have pairs. Okay, all base pairs have pairs. I don't know if that's grammatically correct, but anyway, you can now see the difference here between sticky end and blunt end. So meaning to say, ang blunt end no overhangs. Excuse me, naugan akong tutunan. So, again, majority of the recombinant DNA are actually using the sticky end. Though na apoy, I'm not really into the blunt ends, pero din hita sa sticky ends. So, mas familiar ko sa sticky ends. Okay, so here, target gene, gikat siya using this restriction enzymes, di ba? Naka sticky end, napo ni siya sticky end. Okay, pagkat anak niya, here comes the plasmid here. Okay? Gitangtang ni nimo kini siya mao man siya ang target gene. So gikat ni nimo, imuha ning gikuha. Okay? Imuha ni siyang gikuha and then imuha siyang gi-insert sa plasmid. Take note nga ang plasmid is gamiton nimo the same restriction enzyme kung unsa imuhang gigamit dinhi sa katong mga quote unquote donor nga genome. Okay? Kay aron ang iyang sticky end here sa imuhang target gene will correspond to the sticky end of the plasmid so that they can be recombined. Okay? Again, it is necessary that the restriction enzymes nga imuhang gikat sa imuhang usa ka genome will also be aron makuha nimo ang target will also be the same restriction enzyme nga imuhang gikat sa plasmid so that ang ilahang ends will be the same aron naakay point where they can be successfully attached. Okay, so I hope that is clear. Okay, so this is now the restriction enzyme nomenclature. Okay, I'm not going to include this one, but just to give you an idea how they are named. So say for example, eco R1. Okay, so that's actually um, Escherichia coli, the source sa restriction enzyme. Remember ha nga ang restriction enzymes they are sourced from bacteria. Okay, E. coli and then unsa siya nga generation and then unsa siya nga type eco R1. Okay? Kini pong TAC1, gitawag siyang TAC1. That's Thermos aquaticus type 1. Okay? Ang R1 that's this the kuan Escherichia coli ang R sa base na siya sa kung unsa siya nga strain sa E. coli. Kani siya hindi 3 Haemophilus influenzae and I was able to use this one as well as this BAM H1 when I was still studying my master's degree. So hindi 3 Haemophilus influenzae type 3, di ba? Natoy type depending on structure, depending on katong ihang site kung asa siya kan. And this is the sticky ends where they can actually be cut. They actually cut. Okay? So kana siya is ikat na niya. Okay? Ang BAM H1 Bacillus amyloliquefaciens. Okay. Nindot ning tesa kay na ini labay nga helicopter. Okay. So amyloliquefaciens. So man gitawag tang twister ang scientific name. and nakay AL1 or the Arthrobacter luteus. Okay, so this is kani blunt end nilang makuha. Kini sticky ends, kini siya is blunt end. Okay, nakuha ninyo? Nakita na ninyo, di ba? Okay. So, how do kato kung mention how do this restriction enzyme nomenclature works? So, ang E kato sa eco R1 or eco R E is equals is Escherichia coli. Okay, the genus species, the strain, and the first identified order or identification. Mo tong nakakuha ka og echo R1. Okay, so mo to kung mention ang R signifies the strain of that bacterium. Okay, so palindromic sequences. Take a look at this one here. Palindrome siya. Kung unsa ang padulong sa left, maupod na ang padulong sa right. Okay? So that is where restriction enzymes actually cuts. 'Di ba? So say for example kini, cut siya dinhi kay palindrome man ni siya. Palindrome padung sa left, padung sa right. Na, that's palindrome. Another one is here, palindromic. The difference between palindrome and mirror repeat. So palindrome siya, kini siya padulong din ha, kani siya is padulong din ha. So pwede na mo cut ang restriction enzyme din ha. Okay? So Hindi 3 is here. How Hindi 3 works. So sa palindrome nangita siya palindrome, take note ha nga identified na siya, and then cut siya din ha. That's why naakay sticky ends here. Okay? So mao na ni siya nga once you are able to cut the genes na kay recombinant DNA, pwede nga take note ha nga ang plasmid can carry different segments. So pwede na nga makakarry siya og 2 or 3 or 4 different genes coming from different sources and then i-recombine as one. Mo na nga nanakay rDNA, particularly plasmids. Okay. So when you say recombinant DNA cloning, take note nga recombinant DNA cloning must be copied, multiply, or clones to ensure that the product can be used multiple times. So say for example, kini, 'di ba, i-insert man ni siya sa bacteria kining mga recombinant DNA. So, ang imong buhaton ana as genetic engineer aron to preserve. Say for example, pila ni ninyo ka months gi-work aron ma-insert ni siya na kay recombinant DNA. Okay? And then imuha lang pasagdan nga mawala. So ang imong buhaton is that you have to clone it. So ang pag-clone, pag-insert na nimo sa bacterium, take note nga ang bacterium must be living, and then naa magyunay cell division, 'di ba? Kaning ilang binary fission man na sa kaning mga bacterium. So nay binary fission, so it will divide, 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 then you now have the clones. So ang katong uban nga mga copies, okay, katong uban nga mga copies, pwede na to na mo store, and then you can go back sa kana, pwede i-store na nimo for many years, and then by the time make sure lang nga naa kay kanang record, and then by the time nga mag-study na pud ka, then all you have to do is to get some samples of bacteria that are recombinant, that carries recombinant DNA. So note that recombinant DNA is mostly placed through plasmids in bacteria. Hence, cloning is through binary fission. So that's what I was telling you a while ago. Okay. So here another kini DNA cloning. Okay, so eukaryotic chromosomes insert nimo din ha, and then ni-multiply na siya. Okay, ni-multiply. So you are actually producing lines of different generations, because even though nga kapila ni sila mo kuan, as long as there's no mutation sa plasmid, even though kapila na siya mo binary fission, it will still carries the same nga genetic sequence nga imuhang gi-insert sa plasmid. Okay, so we now go to DNA libraries. When you say DNA libraries, these are techniques in storing genetic information. And there are two types of genetic or DNA libraries. First is the genomic DNA library, and the second one is the complementary DNA library. Okay, and we are going to discuss those ones briefly. Okay, so when you say genomic DNA libraries, they are collection of different desired DNA fragments stored in replicating vectors. Stored DNA segments are cataloged so that it can easily be accessed when needed. So, for example, you find an emo stored and analyzed. Example for that are recombinant DNA clones kato atong gigamit nga genomic. Okay, so here this is now genomic DNA library works. Okay, so gi-isolate nimo ang DNA from cells, i-digest nimo with restriction enzymes, gi-cut nimo, gi-cut, and then nakay mga plasmids gi-recombine, gibutang sa bacterium, okay, and then gipadaghan siya. So this is now a genomic DNA library. Okay, and during my master's degree, wa gid ko ka kuan ani, I was not successful because naabtan ko sa time. Okay, but we are actually going into this direction unta. Okay, and then siguro sa napulo mi kabuk sa labad to, there was only one who was able to successfully clone or yeah, successfully make a genomic DNA library. But anyway, I've learned some lessons on those experiences. Okay, what about these complementary DNA libraries? So DNA copy of an mRNA molecule generated by reverse transcriptase. So complementary DNA. So lahi na ni siya ha. So DNA, ang complementary DNA is DNA copy of an mRNA molecule generated by reverse transcriptase. Okay? So I know nga na mo idea reverse transcriptase asa man na, 'di ba? Katong sa pag-detect whether you are positive sa COVID or not, 'di ba? RT-PCR, reverse transcriptase. Okay? So remember that mRNA is exactly the reflection of DNA segment, 'di ba? So going back into the katong gitawag nato nga katong central dogma, 'di ba? From DNA gikopy siya a segment lang yun sa mRNA transcription and then protein translation. So ang nahitabo ani is that ang gi-store nimo is just the mRNA, because anyway ang mRNA you can reverse that one to produce the DNA. Mo na gitawag to reverse transcription. Okay? But you need a specialized enzyme known as reverse transcriptase for that to happen. Okay? So from DNA to mRNA, pwede na kag makahimo from mRNA to DNA. Okay? So it is just a representative, but the downside is it is just a representative of a portion of a DNA sequence, because mRNA are much smaller compared to the whole genome. So kung unsa ra ang content adtong gitawag natong mRNA is maora pud to ang iyahang ma-produce nga DNA. Okay? So maora po na ang iyang downside. But anyway, it is still very much applicable, okay, sa sciences. Okay, so this is how a complementary DNA library forms. So say for example, you have here tissue or cell, gi-isolate nimo ang RNA. Okay, reverse transcriptase, say for example here you have an RNA, you now have instead from DNA to RNA, you now have RNA to DNA. So mo nang complementary DNA. So take a look at this one, single strand nahimo siyang double strand, pero mao lagi to kung unsa ra kataas ang imong RNA dinhi, mao ra po nakataas ang imong DNA, ang imong complementary DNA. So from this complementary DNA, okay, imuha nang gigamitan og katong giligate nimo. So when you say giligate, is gamitin mo restriction enzyme, di ba? Kay ang kining RNA nimo di man nimo ma-insert sa imong plasmid, di ba? Kay single strand ya, double strand. So imuha sang i-convert into DNA, then from DNA gi-cut nimo through restriction enzyme, gi-insert nimo dinha, then transcription and growth, and then DNA isolation. Gi-isolate nimo. Meaning to say, kung enzyme mo gigamit dinhi, mao po na ang sequence dayon is katunang nag-carry sa complementary DNA. Okay? So, I think, klaro to. So, dinhin na ta sa kaning gitawag nitong CRISPR. And kining CRISPR, this is actually a type of restriction enzyme, but it is more accurate compared sa katong mga contemporary nga mga restriction enzyme, di ba? Nakatong mga HEND3, EcoR1, those are restriction enzyme. CRISPR is another technology using restriction enzyme, pero they are still molecular scissors, pero they are very much accurate compared to contemporary nga mga traditional nga restriction enzyme. So CRISPR stands for clustered regularly interspaced short palindromic repeats. Again, nigawas na po ni siya ang palindromic because as of now dinha natong understanding that restriction enzymes can cut through palindromic na mga sequences. So when you say CRISPR, they are family of DNA sequence from prokaryotic cells which aids in bacterial defense. So ang kaning CRISPR, kaning nga kuan, they were discovered nga ang bacteria is naay capable of defending themselves using kaning nga principle. Okay? So it is more accurate than traditional restriction enzymes, and they were discovered by Emmanuel Carpentier and Jennifer Doudna, which won the 2020 Nobel Prize for Physiology and Medicine. Because if you can just dig deeper into the nature of this CRISPR, you would notice nga there is an immensely huge or mountains of possible nga application sa sciences that can benefit the entire humanity, the entire society. So how do CRISPR works? So here kani Cas9 protein no na CRISPR ang uban na Cas9 mao na Cas9 protein forms a complex with guide RNA in a cell. Kani siya mao ni ang gina-engineer gyud, and then small segments lang. So this complex attaches matching genomic DNA sequences adjacent to a spacer. Kani siya. Okay? And then the Cas9 RNA, kining Cas9 cuts the double strands of the DNA. Gikat niya, okay, and then program DNA may be inserted at the cut. So, ang CRISPR, kaning Cas9, okay, iyahan ang gikat, and then naa kay gustong i-insert nga DNA, pwede na siya. Okay? So, meaning to say, since na siya gi-insert nga DNA, as okay, remember nga ang nag-carry ani pwede siya matawag nga chimera, and at the same time the organism carrying inserted DNA are already mutants because mutants na siya because mutants are defined as any organism carrying katong mga genes nga na-alter. Okay? Mana siya. Okay, so let's go now to the transgenic organisms. Okay? So once organisms were genetically engineered or edited, they can now be referred as genetically modified organisms or kanang gitawag natong GMO. And there are just a lot of people who are against GMO, di ba? So may isa nga dili ta ni GMO, sa example kanang sa mga corn na may common may ng corn, wheat, unsa pa kanang mga flowers or kanang mga food, even sa kanang mga fishes or and other nga mga animals. Okay, ni siya nga dili ta nag GMO kay makadaot sa lawas. Siguro, pero as of now, wala pa'y solid nga evidence nga mga ka-points ato that consuming GMO products can actually harm our health. Okay? Though na of course na mas like freedom to resist consuming GMO products. Okay? Kana siya. Pero the funny thing is daghan ang moingon nga dili GMO, dili sa GMO, pero was lang kibalong ilang gipang kaon GMO na. Okay, kana siya. So, when you say transgenic organisms are those genetically edited where desired genes from sources are incorporated into their genome. So, say for example, transgenic bacteria, animals, and plants. Katong atong gi-mention ganyan, if ang usaga ni ka-organism are carrying rDNA, ito katong recombinant DNA, mana mga transgenic organisms na sila, mutants. Okay, so, kini siya. So, unsa may application ani niya? So, say for example, gene for milk. Okay? Na kay foreign gene, regulatory region, gi-insert nimo. Now, take note nga usara na siya ka genome pero pwede na nimo ma-insert sa different groups of animals. So, say for example, ang isa lang ka katong kining isa lang ka recombinant rDNA, pwede nimo ma-insert dinhi sa kuan, sa goats, sa mice, what else? Rabbit, pigs, goat, sheep. And then naakay kani siya, kaning cow, di ba? So more milk ang mogawas sa ilahang mga progeny na dili sila pero sa ilahan na ng progeny sa ilahang mga succeeding nga mga generation. So mao na nga kani siya if ever nga kining cow is mo produce lang og say for example in one day 1 liter, since nga na siya gi-enforce iyang gene for milk protein. Okay, so siguro after anak niya is makaanak na siya, then it can produce 10 liters na, from 1 liter to 10 liters. So na enhance ang imuhang food production. Okay, so ina na siya, that's basically the idea there. Okay, so say for example, kini, I don't know if you happen to watch this film. So I was able to watch this film. Kaning gitawag to Rampage. Okay, so ang sa Rampage, they are actually using the transgenic animals. So say for example, the wolf, nam kini kani. This is na ay dire day ang wolf kini kay ang crocodile man ni and kini kalimot ko kinsa ani albino mo ani ang friend, di ba? Ang wolf. So say for example, ang wolf is ang wolf is porcupines, na siya traits for porcupines kini iyang kuan, kini iyang quills. Lemur, nahimo siyang lemur, 'di ba katong kabantay mo sa katong building nga nilupad siya. So actually those are lemurs and genes for I don't know, di ini gikuha nila ang hypersized genes. Okay? So basically, the recombinant DNA technology can actually theoretically can produce new organisms. Theoretically, nga. Say for example, mapakakita og isda nga mulakaw sa yutha or naipako pa like birds, pero isda siya. Theoretically, you can actually do that. Okay? Pero, I don't know kung gi-apply. Siguro, na mga trials and tests, pero wala pa siya ma-produce. But theoretically, it is possible. Okay? So what else? So what do you think is the future application of kaning gitawag natong recombinant DNA? Okay, man naman ini. So future application. Okay, so do you think nga we can have superfoods nga one bite, okay, it will last for 1 month, di ba? Or one bite lang, kaning usa ka kutsara lang is naa dito ang imuhang vitamins, minerals, na dito ang milk, na dito ang iron, na dito one bite lang, usa ka kutsara lang sa kanon, then you have now a complete food, di ba? So it can be applied to better the humanity or it can be applied to destroy humanities, di ba? Sa mga tatong gi-mention before, di ba? So it actually depends kung unsa ang inyong preferred nga application sa ato ang technology. Pero mao lagi na nga kanang gitawag nato nga um, you as a scientist, siguro some of you might reach that level nga genetic engineer yun mo. Okay? So isulod yun sa inyong mind nga science should benefit humanity. It should help build humanity, not to destroy humanity. 'Di ba? Okay. So sige. So I think din ra ni end ang ato ang discussion. Okay? So you can actually dig deeper into the nature of recombinant DNA technology. Okay? So don't just stop here. Don't just stop sa ato ang lecture. Don't just stop sa inyong mga handouts. Go further into to being informed about the subject matter. Okay, so kana siya. So thank you very much. And if you have any questions sa ato ang discussion, don't forget nga mo comment ka or you can send private message ako. Okay? Or comment din ha sa ato ang section, sa atong comment section, put your questions sa atong comment section and I will be happy to respond to the best of my ability. Okay? So daghang kay salamat and enjoy your day and be happy, stay in love and be a good citizen. Bye bye.